Good morning, everyone. I'm Laura. I'm a molecular biotechnology at Vanvitelli University in Italy. And today I will talk with you. I will have a lesson about schizophrenia. I will introduce this disease, and after I will talk with you about the glutamatergic hypothesis. Finally, I will do also a very short parenthesis about biomarkers and last evidence of the link with the D-aminoacids. So this is my lineup to show you how I will do my talk. I decided to divide my speech into three parts. So first of all, I will give you some epidemiologic and pathophysiologic data, and after I will talk about a glutamatergic hypothesis that will be the main topic of my talk, and finally I will show you last data in the link between the amino acid and schizophrenia. So let's start with a very short introduction about schizophrenia in order to a better understanding something of this disease. Schizophrenia derives from two Greek words that are schizei, that means to split, and phrenos, that means mind. So it refers to a split mind, the separation of the mind from the reality. The separation of the mind in cognitive processes, and it is a very complicated mental disorder characterized by a breakdown of thought processes and behavioral functions. Let's see some epidemiologic data. So, schizophrenia is a severe brain disorder affecting 0.3% or 0.7% of population worldwide. In this image, you can see the distribution of the disease, and in red, we can see more person affected by schizophrenia, of course. This disease is diagnosed 1.4 more frequently in males than females, and the age of onset is around 20s in males and between 25s or 30s in females, while in childhood is rare. Okay, so in this slide you can see some typical anatomic features of the disease. Researchers have consistently found structural aberrants in distributed brain regions with the help of modern neuroimaging tools like the magnetic resonance imaging. For example, there are evidence that describe that the reduction of gray matter in schizophrenic brain is a phenotype of the disease. So, gray matter loss is implicated in schizophrenia and is known that it occurs in adolescence. In this image on the top represents a spatiotemporal map of this loss process in a study during 5 years. But the most interesting thing is that this loss of gray matter is directly related with the enlargement of ventricles. In fact, this loss of gray matter induces a ventricular enlargement. That is another typical phenotype of this disease, as well as we can see in this image here in the bottom. Ventricular enlargement is one of the most consistent abnormal structural brain findings in schizophrenia. The ventricles of patients with schizophrenia are approximately 130% the size of the normal controls. In other words, if we want to translate this data in absolute volume, this is a difference in order of 1.5 cube centimeters. That's relevant. The ventricular enlargement is directly related with a shrinkage after the onset of the brain processes associated with schizophrenia. And finally, we have another pathophysiologic feature, typical feature, that is the enlargement of amygdala. In fact, several recent meta-analyses have reported enlarged amygdala in patients affected by schizophrenia. And it's very important because amygdala plays a crucial role in emotions. In fact, the stimulation of amygdala in humans evokes emotions as well as complex hallucinations. All those typical brain aberrants in distributed brain regions are directly related with symptoms of schizophrenia, as we can see in this slide. In fact, we know very well that the brain is the major complex organ in the whole body system. That's because brain regulates both voluntary and involuntary functions. But anyway, schizophrenia is a very complicated disease, characterized by many types of symptoms, as we can see. We can distinguish all symptoms of schizophrenia into three main categories. So we have positive symptoms, negative symptoms, and cognitive symptoms. Positive symptoms are those ones that are absent in a normal subject. Negative aren't usual in the subject, and cognitive, of course, regards cognitive functions. So, those symptoms, cognitive ones, are directly linked with the brain region that is named prefrontal cortex. In this slide, I highlight the prefrontal cortex in orange. So, another important symptom of schizophrenia is a positive one, that is hallucination, for example. And we can imagine that hallucination is directly related with this, so occipital lobe, because this is a brain region that controls visual functions. That is a hypothesis, for example. Or, for example, we have another hidden region that is the amygdala that I talked with you in the previous slide. And amygdala controls emotions, so it is directly related with the negative symptoms, for example. Another important main brain regions involved is hippocampus, that is involved in cognitive functions too. Another important region that I wasn't able to highlight in this image is the thalamus. Thalamus is a very little brain region, hidden, but very important because it controls the coordination of all information they are from the whole brain. So this region, that is the thalamus, is directly related with all schizophrenia symptoms. Okay. And symptoms are very important today to make a diagnosis, because today it is possible only to make a subjective diagnosis by a doctor. To make a diagnosis, the patient must have for 1 month period or longer 2 or more symptoms, including at least one of the positive ones. During a period of 6 months, the patient deals with residual periods, which are characterized by negative symptoms. So we can easily understand how it could be important to have a biological biomarker in order to make an objective and early diagnosis of this disease. But it isn't a very easy work, because we are talking about a disease localized in the brain. So exams could be invasive. And also, what about causes? We don't have a specific causes of schizophrenia. That is another point that makes more difficult to have a biomarker or make an objective diagnosis. Today we don't know the causes of schizophrenia, but we have many hypotheses, and we can distinguish these hypotheses into two main hypotheses. So we have a neurodevelopmental hypothesis and biological hypothesis. What about a neurodevelopmental hypothesis? We can say that has been for longer the main hypothesis. It regards the neurodevelopment and suppose that it can be affected by two types of risk factors. We have genetic risk factors and environmental risk factors. But the genetic component of schizophrenia is high. The heritability of schizophrenia is about 79%, with a shared influence of the environment of 11%. Both genetic and environmental risk can interact during pre- or postnatal period to induce symptoms of schizophrenia in the early adulthood. In fact, synaptogenesis. First of all, I want to focus your attention on this image. This is a flowchart about the brain development, in which one there are highlighted two critical phases in brain development. That is, one is a prenatal phase and another one is a postnatal phase. So we have a prenatal phase regarding the development of the brain and a postnatal phase regarding adolescence. So, synaptogenesis is established during the second and third trimester of pregnancy and continues during childhood. In a proposed two-model HEAT, early prenatal insults, both genetic or environmental factors, may lead to a dysfunction of neuronal networks and a vulnerable status, while a second hit during a critical brain development period in adolescence may induce the onset of the disease. To develop this two-hit model, they made many studies in family, twins, they made adoption studies, and finally they provide the evidence of the contribution of genetic factors to schizophrenia risk is estimated around 60 or 80%. Genetic risks are structural changes of the genome, environmental interaction, epigenetic factors that are very important. Environmental risks are many, but the more interesting are obstetric complications and also cannabis use. For example, it has been evaluated that cannabis is associated with a large risk of schizophrenia and spectrum disorders if the cannabis use starts before adolescence or during adolescence, so before adulthood. That's very important and very interesting. What about biological hypotheses? Biological hypotheses instead are more recently. They regard neurotransmission, in particular chemical synapses. That because I choose this sentence, a neurochemical approach. We are talking about a neurochemical approach. The basic neurochemical hypotheses of schizophrenia include the dopamine, the glutamate, and serotonergic. But today I will focus on the glutamatergic hypothesis. Glutamatergic hypothesis that regards glutamatergic synapses. But why today we are investigating on glutamatergic hypothesis? Every time in science, things and studies starts from the first step. The first step is always the same. The first step is the observation. In fact, they observe that the use of some antagonists of NMDA receptors like ketamine or phencyclidine, that are antagonists of NMDA receptor, induces schizophrenia symptoms. So, they decide to investigate the glutamatergic hypothesis. Ketamine in healthy volunteers produces a thalamic disconnectivity pattern that are resembling of the thalamic disconnectivity pattern they saw in schizophrenia. In particular, ketamine produces thalamic hyperconnectivity with several cortical, sensory, and motor regions that are similar to those who are reported in schizophrenia. Experimental studies have demonstrated that ketamine can produce behavioral responses in rodents. And they see also that in healthy human volunteers, um, if they assumed ketamine or phencyclidine, they manifested symptoms very closely resemble the positive, negative, and cognitive one. What happened? So in schizophrenia. That's because they decide to investigate on glutamatergic hypothesis. But what is the glutamatergic hypothesis? Today, glutamatergic hypothesis is considered one of the etiologies of schizophrenia, and it regards glutamatergic synapses and glutamatergic neurotransmission. But in order to understand how it works, I want to show you a video. How can I show you this video?
Glutamate is the most abundant excitatory neurotransmitter in the brain. Release of glutamate is essential for normal function of neurons, but the levels of this neurotransmitter must be tightly regulated to avoid toxic effects on neurons. Let's take a closer look at normal glutamate signaling. As an action potential propagates into the terminal of a presynaptic neuron, vesicles storing glutamate fuse with the cell membrane and release glutamate into the synapse. Once released, glutamate can bind with several types of receptors, including postsynaptic ionotropic and metabotropic receptors that are responsible for a range of downstream effects, including initiating rapid communication between neurons, releasing brain growth factors such as BDNF, and modulating synaptic plasticity. After its release, glutamate is cleared from the extracellular space through excitatory amino acid transporters, or EAATs, which are located primarily on the surface of glial cells. Once glutamate is inside the glial cell, it is converted to glutamine, which no longer possesses excitatory properties. Glutamine is then shuttled back into the presynaptic neuron to be recycled into glutamate. Dysfunctional glutamate signaling can negatively alter brain function. Alterations such as increased release of presynaptic glutamate, the direct release of glutamate from glial cells, or an impaired ability to clear glutamate from the extracellular space can lead to excessive excitation of glutamate receptors. Too much glutamate receptor excitation can have neurotoxic effects and is implicated in various diseases, including ataxias, ALS, neurocognitive diseases including Alzheimer's disease, and neuropsychiatric disorders including mood and anxiety disorders.
So we see that glutamate synapses work with glutamate. Glutamate has both an amino acid and a neurotransmitter natures. But today we know that glutamate is the major excitatory neurotransmitter in the nervous system. In this image, we can look how we can get glutamate. Reality is that we have glucose. Glucose is able to cross the blood-brain barrier, so it enters in glycolysis, and after in the TCA cycle, and after alpha-ketoglutarate. In alpha-ketoglutarate, we have the adding of amino group, and we have the glutamate. And glutamate from the astrocyte can do many things. So glutamate is involved in many normal physiological functions, and it's also importantly involved in many cognitive functions. Glutamate was known to have a role in central nervous system more than 75 years. but only in 1984 it was truly acknowledged as a neurotransmitter. Glutamate was originally speculated to serve a metabolic function in the central nervous system, as it was found within numerous intracellular compartments, including the cytosol and mitochondria of all central nervous system cell types. However, it is known that despite its ubiquitous nature, levels of extracellular glutamate are indeed tightly regulated. Um, and this tightly regulation is directly related with its neurotransmitter function. So, the tight control of glutamatergic neurotransmission is an energy costly process requiring multiple regulatory processes and high levels of glucose and oxygen consumption. So, like all amino acid, we have an amino group and a C-terminal group, and this is so. Let's go to the next slide. Yes, as all neurotransmitter, glutamate needs a receptor. So glutamate receptors are numerous and highly complex. First of all, we can distinguish into two main categories. So we have ionotropic glutamate receptor and metabotropic glutamate receptor. Ionotropic glutamate receptor has the primary function to allow entry of ion into neurons, while metabotropic receptors need G protein. So they are G protein coupled. We are interested in NMDA receptors. So NMDA receptors are the main category of ionotropic receptor, more involved in glutamate synapses. That's because we are interested in the study of them. So, NMDA receptor play crucial roles in many neurological functions in central synapses. These receptors regulate the structural and functional plasticity of synapses, dendrites, and neurons by activating a series of calcium-mediated signaling pathways. NMDA receptor play an important role in the straightening of synapses, which is known as a long-term potentiation. It is an important neurological process associated with strong memory formation. NMDA receptors are composed of four distinct subunits, two glycine binding, as we can see these in blue, so two glycine binding GLU and 1 subunits, and two glutamate binding GLU and 2 subunits in orange. So these subunits are assembled in a heterotetramic way to form a central cation permeable channel pore. This slide is highlighted calcium like ion, because calcium is involved, and we will see it in the next slide. So, these subunits are assembled in a heterotrematic way, and all glutamate receptor subunits are similar in architecture, so they comprise four domains, as we can see in this image. So we have a large extracellular ATD, a bilobate ABD, a pore-forming transmembrane domain, and an intracellular CTD. Okay, so how an NMDA receptor works? This is very interesting because, like any other receptor, an NMDA receptor undergo a ligand-induced conformational change to be activated. Glutamate release from the presynaptic terminal into the synaptic cleft at very high concentration bind the postsynaptic AMPA receptors that are easily receptors. They are easier than NMDA receptor, less complex than NMDA receptor. In fact, when glutamate binds AMPA receptors change their conformation and open the channel. So we have the entry of sodium. This massive entry of sodium determines depolarization of the membrane. This depolarization of the membrane determines. I forgot to say something very important in this slide. So this is the NMDA receptor. NMDA receptor to opens needs both an agonist action, that is the glutamate, and a co-agonist action, that is the glycine. But also they have a binding site for magnesium. When magnesium is binded with the receptor can't bind glutamate, so it can't open. It can't open. But when we have the depolarization of the membrane, magnesium, we have the release of the magnesium, so an NMDA receptor can bind glutamate, and by the binding of glutamate and glycine, we have the opening of the channel. And when the channel is open, we have that calcium across the barrier, the membrane. And the accumulation of calcium determines second response. So postsynaptic response are mainly regulated by calcium. And we have all what happened in postsynaptic cell. Okay, let's see glutamate pathways in order to understand all it happens in the brain. So, glutamate pathways are linked to many other neurotransmitter pathways, and glutamate receptors are found throughout the brain and spinal cord in neurons and glia. In this image, we can see glutamate pathways in red, and we can see GABA pathways in green. It is important to understand that glutamate pathways and, for example, GABA pathways interact. And they are involved in many brain regions, for example, in prefrontal cortex, in accumbens, in thalamus, in amygdala, in hippocampus. And I choose also this image in order to understand how those regions are involved in glutamate pathway. No? So let's see something more difficult. So being aware of the complexity of the situation, I try to explain in a simplified way one of the possible pathways they can occur. So, hypothesize that the neuron A is a glutamatergic neuron, the neuron B is a GABAergic neuron, and the neuron C is another glutamatergic neuron. So, neuron A and neuron C as a glutamatergic neuron produces glutamate, while neuron B produces GABA. Glutamate and GABA has two different functions. For example, glutamate activates. So glutamate has an activatory function, while GABA has an inhibitory function. So if neuron A produces GABA, we have the activation of this neuron that can produce GABA that inhibits the neuron C. And this is one of the possible pathways. Yeah, it is a physiological pathway. But what happens if we have an hypofunction of NMDA receptor, that is a glutamate receptor? If we have the hypofunction of NMDA receptor that is localized on GABAergic neuron, we have that this neuron can produce GABA, so can't produce its inhibitory function on the neuron C. So we will have the hyperactivation of this glutamatergic neuron. And this hyperactivation of the glutamatergic neuron is what happened when we administered a ketamine and phencyclidine to normal unaffected volunteer. That's because it's important. So now we are entering in the third part of my talk. So we are seeing that last studies conducted on postmortem brain revealed the presence of the amino acid. So there has been done some researches about the role of the amino acid in brain function, looking for a link with some brain disease like schizophrenia, for example. In particular, I show you that NMDA receptor need both an agonist and a coagonist to opening the channel. So, a relevant study found that the amino acid are able to activate this channel because the D-aspartic acid can work as an agonist by binding the receptor in the same site of glutamate. And the serine contribute the opening, acting as a co-agonist by binding the receptor in the same site of glycine. Normally, glutamate as a neurotransmitter is tightly regulated to avoid the glutamatergic neuron hyperactivation. So, these findings is relevant and plays a crucial role in future researches. Also because maybe the amino acid or other connected molecules in future could be used as a possible biomarkers due to make an objective diagnosis. That's because it's relevant. What about the So we see that the aspartic acid is able to bind the glutamate site. This is able to bind the glycine site. But why we are interested in this Because this is an endogenous ligand. Endogenous. In fact, we can see there that this came always from the glucose. So glucose across blood-brain barrier, entry in glycolysis, and we obtain L-serine. L-serine is converted in D-serine thanks to an enzyme that is the racemase. So racemase converted L-serine in D-serine, and D-serine is inside vesicles. So we have the rejection of vesicles in the synaptic cleft, and we have that this serine bind NMDA receptor. And there exists many evidence about the role of this serine that is a relevant role. In fact, this serine is one of the naturally occurring proteinogenic amino acid synthesized in the human body from other metabolites. This serine is synthesized in the brain. This serine is involved in NMDA receptor.